The first stage of cellular respiration is glycolysis. Glycolysis is split into two phases, the energy investment phase and the energy payoff phase. In the energy investment phase, 2-ATP is used to transform the glucose from the food into 2-ADP and 2-phosphate. In the energy payoff phase, the products from the previous phase, the 2-ADP and the 2-phosphates, are used to form 4-ATP, 2-NADH, 2 hydrogen ions, 2 pyruvates, and 2 water molecules.
After glycolysis, pyruvate in the cytosol enters the mitochondrion by an active transport. Inside, enzymes catalyze 3 steps. First, the carboxyl group oxidizes and releases CO2. CO2, or carbon dioxide, then oxidizes and forms acetate. Lastly, coenzyme A attaches to the acetate to form acetyl-CoA, which is then used in the citric acid cycle.
In the process of the Krebs cycle, acetyl-CoA adds its 2 carbon acetyl groups to oxaloacetate, producing citrate. Citrate is converted to its isomer, isocitrate, by removal of one water molecule and addition of another. Isocitrate is oxidized, reducing NAD+ to NADH. Then the resulting compound loses a CO2 molecule. Another CO2 molecule is lost, and the resulting compound is oxidized, reducing NAD+ to NADH. CoA is displaced by a phosphate group which is transferred to GDP, forming GTP, a molecule with functions similar to ATP. GTP can also be used, as shown, to generate ATP. 2 hydrogens are transferred to FAD, forming FADH2, and oxidizing succinate. Addition of a water molecule rearranges bonds in the substrate. The substrate is oxidized, reducing NAD+ to NADH, and regenerating oxaloacetate. And that is the Krebs cycle.
The last stage of cellular respiration is oxidative phosphorylation. The first part is the electron transport. In this stage, NADH produced by the Krebs cycle travels through an electron transport chain of multi-protein complexes, being oxidized and reduced. One source of electrons is from the FADH2. During this process, the amount of free energy slowly drops into a series of smaller steps that release energy into manageable amounts. This process also produces water. ATP synthesis is then continued in chemiosmosis.
In the final stage of oxidative phosphorylation, known as chemiosmosis, there is an enzyme known as ATP synthase. In chemiosmosis, energy stored in the form of a hydrogen ion gradient across a membrane is used to drive cellular work, such as the synthesis of ATP. ATP synthase uses this energy to power ATP synthesis. Hydrogen plus ions drop into ATP synthase to form ATP from ADP. So ADP is adding a phosphate group to produce ATP.